Good morning, good afternoon, or good evening. My name is Bert Hofman, and I have the honor to chair this symposium of the HOPE program, the Harvard-Oxford program in epidemiology. It's a symposium on developments in neuroepidemiology. Great to have you all, and it is my pleasure to first give the floor for a few moments to Dr. David Hunter, who is the auctor intellectualis and the prime mover, director also of the HOPE, the Harvard-Oxford program in epidemiology. David, please go ahead.
Thanks, Bert. So the aim of the program is to bring the best of Harvard epidemiology together with the best of Oxford epidemiology. So it's a great pleasure to be here in the symposium. Very much looking forward to it. Thanks to all the speakers. And I think we're going to learn some very interesting things. So thank you.
Well, David, thank you very much. Indeed, we have a symposium occasionally, but nearly every week we exchange our seminars, both from Oxford and from Boston. So everybody very much invited there as well. With that, it's my great pleasure to introduce the first speaker. Dr. Alberto Ascirio had his MD, graduated in medicine from the University of Milan in Italy, and joined the faculty of the Harvard School of Public Health in 1992, and is, may I say this, a world expert in neuroepidemiology, particularly focusing on multiple sclerosis, on Parkinson's disease, on ALS. Alberto, it's wonderful to have you after, I think, a great contribution early this year in science, and you will talk about that because you will speak about multiple sclerosis is a rare complication of Epstein-Barr virus infection. Dr. Alberto Ascirio.
Thank you, Bert and David. So let me get my screen on. Let's see. This one. Okay, can you see my screen?
Yes.
Okay, thank you so much. Well, I'm really honored to present to this group. This is a work we started over 20 years ago. And in fact, David, I don't know if you remember, but you were also one of our first paper on EBV and MS. So let me say just a couple of words on MS. It was beautifully described by Charcot in the 1800s in these pictures. So it's a demyelinating disease in which you have demyelinating lesion in different parts of the brain and spinal cord. So the symptoms can be extremely variable depending on where the lesions are. In most cases, start with a relapsing remitting course that is followed by a progressive phase. It's a lifelong disease and can lead to serious disability. So if you look at the pathology, it's consistent with autoimmune mechanism. But the role of infection cannot really be excluded when you look at the pathology, the features of the pathology. But the etiology until now has been described as unknown. So it's considered the most common disabling neurological disease in young adults. There are about 2.8 million people in the world with MS. The incidence is mostly in young adults, and lifetime risk in women, which is higher than in men, is about 1 in 200. So there are currently very good treatment for MS that suppress the disease activity but do not achieve a cure. And the recent study found a rather high cost of MS in the US at 85 billion per year. The idea that infection could be related to MS has been proposed for over a century, often without any data, generating widespread skepticism. In fact, it was a topic that was difficult to approach when I started working on this because people were dismissive after so many false signals. And the link to EBV specifically was first suggested in the 1980s. Based mostly on the similarity between the epidemiology of mononucleosis and MS. So a bit over 20 years ago, we started then to follow that link we did a case control study nested within the nurses cohort. And, this was work by Miguel Hernan, who was a doctoral student working with me at the time. And as you see, infectious mononucleosis stood out among all the other infections, the only one who had a strong association with risk of developing MS. This was a retrospective case control studies with all the limitation. We also look at breast cancer controls to control for possible recall bias, and this is the laboratory confirming mononucleosis. Now, these findings were then reproduced in numerous studies, and in fact, confirmed even in a longitudinal analysis based on the Danish MS cohort. So I think it's a 2-3-fold increase in risk associated with history of mononucleosis. Now, mononucleosis is a quite peculiar disease because it's a manifestation of a primary BV infection when it occurs after early childhood. So the epidemic curve is very narrow, as you see here. By the age of 2022, virtually all people are infected with BV, about 95%, and that is why the incidence of mononucleosis goes down. Now, MS, as I say, the peak of MS is between 20 and 40, about the late 20 here. So at this point, you have very few people who are not infected with BV. Another characteristic of EBV is that the age of infection is strongly correlated with socioeconomic development and hygiene condition in childhood. So if you look at the top, when you look internationally in children between 4 and 6 years of age, in developing countries, virtually all the children are already infected. So mononucleosis is extremely rare. Whereas if you look at France, Sweden, England, so we're around at 40–50% of the children are infected, and then the remaining will remain at risk of infection at later ages, and thus mononucleosis. Interestingly, within the US at the time, these are data from the 1970s, there was a gradient between the Northwest and Southeast, possibly reflecting differences in socioeconomic condition and hygiene even within this country. So when you put this data together, a common explanation was the so-called hygiene hypothesis. This hypothesis was in fact originally formulated in the 1960s and predicted that low exposure to childhood infection, generic low exposure, including parasitic, viral and bacterial disease, would prime the immune system toward more pro-inflammatory immune responses and increase the risk of autoimmune disease in general, and particularly the risk of MS. So according to this hypothesis, you would have expected that individuals with high hygiene in their childhood would be at high risk of mononucleosis and MS. So the link between the mononucleosis and MS would be due to confounding, and there would be no causal association. And so in this case, anything done to prevent EBV infection and risk of mononucleosis would do nothing in terms of affecting the risk of MS. Now, this hypothesis, if true, would have an interesting prediction. That is, those individuals who escape EBV infection in childhood and are EBV negative because of the high hygiene in their upbringing, they should have a high risk of MS, right? Because that is what the hypothesis predict. Now, paradoxically, when you look at the collection of all the case control studies that were done with many limitations, but they suggested that if anything, the risk of MS in EBV negative individual was extremely low. So this paradox, this data from several years ago caught our attention, and we think, can it be that in fact, how do we explain if you go from being EBV negative, you have an extremely risk of MS, but you have history of mononucleosis, you have extremely high risk of MS, and this individual come from the same high hygiene set. So this was the rationale for thinking that there may be an alternative explanation that EBV is in fact the cause of MS. And as I mentioned, that finding is very robust, but this finding was very questionable because it was based on prevalent MS cases, individuals who had MS for 10, 15, 20 years. So we didn't know whether the the EBV positivity in MS cases was in fact due to individuals with MS being more susceptible to acquire EBV. So this was the background of the new study that I'm going to present. The objective were to determine whether EBV infection preceding time the clinical onset of MS, whether it preceding time the elevation of serum neurofilament that was chosen as the initiation of the pathological process. This is a sensitive biomarker of neurodegeneration, and also to explore the potential role of other viral infection or immune dysregulation that was often proposed to explain the link between EBV and MS. So the reason we use serum neurofilament is that there is a pre-symptomatic prodromal phase in MS. So demonstrating that EBV precedes the clinical onset is not sufficient to demonstrate that it precedes the onset of the pathological process. And we had previously demonstrated that up to 6 years before the symptom of MS, there is an elevation of neurofilament in individuals who will go on to become MS cases as compared to controls. So the challenge, of course, was to do a study on a nearly ubiquitous exposure. I understand you covered this topic a couple of weeks ago in this series of seminars. So 95% of individuals are EBV positive, which makes it difficult to find a large population of individuals who are EBV negative and follow them prospectively. So this was made possible by the Department of Diffuse Serum Repository. Over 60 million serum specimens collected from 10 million young men and women in active duty in the US military. And the onset, the occurrence of MS in this population is documented by the Physical Disability Agency because MS is considered a medically disqualifying condition. So individuals suspected of having MS go through quite a rigorous clinical assessment. And the population is very young, which is important because as I show the infection with EBV, the prevalence of EBV positivity increases with age. And if you go too far off past the age of 20, you will have very few individuals at risk. Study design was conceptually very simple. So this is a nested case control study with risk set sampling. So for each of the MS cases, we selected 2 controls with blood samples collected at the same time, matched on sex, age, race, ethnicity, the date of blood collection, and the military branch. The EBV status was assessed by a combination of ELISA and Western blot, and the data could be collected from 801 patients and 1,566 controls. We always use this sort of pretty rigorous procedure when we do any biomarker study to avoid any possible bias. And the Western blot, were quite clear. So this is just a random example, one of the curves. So individuals who were negative for EBV were negative for all the antigens. And those who were positive for multiple antigens. So there was no really ambiguity in deciding who was EBV negative and when these people became infected and EBV positive. Now, for obviously observational study, there is concern about possible confounding. So we chose as a negative control cytomegalovirus, which is also an herpes virus like EBV, transmitted through saliva that displays similar socioeconomic and racial ethnic gradient and disparities in agent infection as EBV. Okay, so let me get to the results. So there were 801 MS cases documented in this population. So when you look at baseline, 6.8% of the individuals were EBV negative, which is consistent with the NHANES and other data that from other population within the US. Among those who were EBV positive, there were 766 incident cases. Among those who were EBV negative at baseline, there were 35 cases, of which 1 occurred in those individuals who remain EBV negative and 34 in individuals who seroconverted. And all these cases were at clinical onset after seroconversion for EBV. The rate of seroconversion for EBV during the follow-up was about 50% over the entire follow-up. which is a rate about 12% per year that is also consistent with that in other population of young adults. In terms of seroconversion, when you look at the left, you have EBV. So the blue line represents the seroconversion among controls, the rate that I've just mentioned. And the green line represents the seroconversion among individuals who develop MS. And on the right, you see comparable data for individuals where EBV and CMV negative. As you see, the seroconversion for CMV were similar in cases and controls. When we look at this data in terms of hazard ratios, in taking the EBV negative group at the reference, individuals with seroconverted for EBV had a 32-fold higher risk of developing MS than those who remain negative, and those who were positive at baseline had a relative risk of 26 compared to those who were EBV negative. You see on the right, CMV seropositivity and seroconversion were not associated with an increased risk of MS. In fact, being positive for CMV at baseline was associated with a modest reduction in MS risk. Now, again, confounding is considered one of the major issues in observational data. Now, to explain a relative risk of 32, and here, borrowing from Tyler Vanderwill E values, so you would need a confounder that has a 60-fold, confer a 60-fold higher risk of developing MS and a 60-fold higher risk of acquiring EBV. And so my argument has been that this is not only we are not aware of any candidate here. It's even difficult to conceive, a factor that would have a strength of association of this magnitude. The strongest risk factor for MS in terms of genetic predisposition is the HLA-DRB1 that confer a 3-fold higher risk of MS. Other environmental factors like vitamin D deficiency, smoking, childhood obesity may double the risk. So we are not close to anything that could explain a relative risk of MS of 60-fold, and even less the risk of acquiring EBV higher, 60-fold higher. Now, perhaps the more subtle explanation is that of reverse causality. So could MS somehow increase the risk of becoming infected with EBV? So that is why we measure the serum neurofilament to establish this temporal sequence, and the antibody titers against the entire human virome meant to assess a genetic dysregulation in individuals with MS that could explain their higher propensity of becoming infected with EBV. So these are the data on NFL. So you see on the left, the level of serum NFL before EBV infection is taken as the reference point. At the time of the first EBV positive sample among MS cases, there is no increase of NFL. It's only in the second sample collected after EBV infection that we see an elevation. So this clearly established the temporal sequence that EBV infection precedes the NFL increase. Among control, there is no relation between EBV infection and NFL, showing that EBV by itself, an individual who are not going to develop MS doesn't affect the NFL levels. Now, the virus scan is a relatively novel technology that allows, with a few microliter of serum, to assess the antibody response to a broad, extremely high number of peptide. You can include anything that you like. Our library included the peptide, 56 amino acid long peptide overlapping by 28, covering the entire known human virome. So all the known human viruses were covered. The peptide are expressed in this phage library that bind the antibody, and then the phage that are not binded by the antibody are washed out, and then you use a massive sequencing by PCR to count the enrichment in which peptide have a stronger antibody response. So I was quite surprised by seeing this data. I thought that this is a rather, noisy, non-specific technique with overlapping peptides across all the viruses. And it was quite striking that all the peptides that were enriched in the cases were virtually all from EBV, with almost no signal coming from any of the other viruses. These are just all the peptides for which there was significant enrichment in either the case in orange or in the control in blue. This was done in 30 MS cases and 30 matched control. Each individual had 2 samples, one collected pre-onset for the MS cases and one post-onset. And matched sample collected at the same time were included for the control that, as before, were matched by individuals of the same age, sex, ethnicity, and branch of the military. So I think that this result basically showed that we are not looking at some generic dysregulation, immune dysregulation, but we are looking at quite specific response against EBV. An interesting feature was that there was often a lag, or in almost all cases, between EBV infection. The blue dot, the first blue dot represents the time of the first EBV positive sample and the onset of MS, which is the orange dot. In virtually all cases, there was only one case, as I mentioned before, that remained EBV negative throughout the follow-up until the onset of MS. So our conclusion was the MS is a rare complication of EBV infection. This, of course, doesn't exclude that other factors modulate risk, including family history of MS and genetic predisposition, history of mononucleosis, a marker of age EBV infection, vitamin D deficiency, tobacco smoking, obesity during adolescence. This is largely work of our group and other over the past 20 years. Now, when you say that, talk about a virus causing a disease in observational study, obviously you're going against the odds, right? There is this conception or misconception that you need an experiment to demonstrate causality. And this was a comment to one of the reviewers when we submitted the paper. So our reply was that when we talk about causation, we mean it in a pragmatic sense. What we say, whether prevention of EBV infection would prevent MS, and this is the concept that underlie all the, most of the achievement of modern medicine and public health. And this, by relying on this pragmatic concept that we've been able to prevent rickets, spina bifida, lung cancer, stroke, poliomyelitis, AIDS, and many other conditions. And I like to make this is the drama of the mortality from lung cancer. These are all data from the UK, where you see the exponential increase. This was an association, of course, in association with consumption of cigarette and tobacco sales, and at the time, Richard Doll and Bradford Hill, published this study in which virtually even among the control, all the control were smokers. But in spite of this, they could demonstrate a significant association. In some way, the data with EBV were in some way similar. Now, EBV causes several diseases. Common is mononucleosis, but causes several complications, including Burkitt lymphoma, nasopharyngeal carcinoma, Hodgkin lymphoma, and other lymphoma, 5 to 10% of gastric cancer, post-transplant lymphoproliferative disease, and others. So it's more the rule than the exception that the common viruses cause rare complication. And I think we now, I'm confident we can add MS to this list. Few words on the virus. It's transmitted by saliva, established lifelong infection in B lymphocytes. It's a latent infection, but is kept really in balance by extremely strong immune response by cytotoxic CD8 T cell and CD4 T cell. And when you have immunosuppression, you get uncontrolled proliferation. So it remain in B cell for the rest of your life, and B cells that are infected with EBV in some way resemble B cells that have been activated by recognizing their specific antigen. This is the B cell receptor. This is the co-stimulatory signal, CD40. They have their downstream activation pathway. And the virus synthesizes two proteins, LMP1 and LMP2, that activate the same pathway that are activated by the B cell receptor and CD40. So in some way, these B cells infected with EBV are immortalized, become non-respondent to apoptotic signal. So we don't know the mechanism. I'll get to that in a moment exactly by which EBV causes MS, but it's interesting that the most effective treatment for MS are based on the depletion of B cell, which are the cytopersistent of the virus. So this is the unsolved question that we are trying to address in the next several years of research. Just calling your attention on the top left here, there is extremely strong association between pre-diagnostic level of anti-EBNA titers, antibodies against the Epstein-Barr nuclear antigen, risk of MS up to 36-fold, quite consistent. And there has been recently a paper in Nature suggesting there is some molecular mimicry between antibody against EBNA1 and the glial protein, glial CAM. This remains to be confirmed, but it's just one of the potential mechanisms. A few considerations that I think are useful in this context, that MS risk is not increased by immunosuppression, so we are not facing a situation of uncontrolled viral replication. In fact, the EBV viral load is only modestly elevated, the strong association I mentioned. The number of infected B cells is extremely small, and in fact, it's much less than the number of cytotoxic T cells that control EBV. All this clearly suggests that the immune response to EBV is causing the disease rather than the virus lytic infection of the virus itself. Also, EBV is a non-oncogenic virus, so we have to consider the possibility of a transformation of B cells that lead to disease. The most effective treatment, as I mentioned, are deplete B cells that are involved not only in antibody production, but also antigen presentation, the release humoral factors that can be related to MS pathogenesis and attract or activate the T cells and the CNS. And there is some evidence of T cells that are dually reactive against EBV and myelin. I think I'll finish with a few words about the implications. So I think in EBV negative individual, a vaccine that confers sterile immunity would almost certainly prevent MS. A vaccine that prevents mononucleosis but does not prevent infection is likely to reduce MS risk, but we cannot be certain until we try. And I think it would be interesting that in EBV positive individuals, because as I mentioned, by the age of 18 to 20, over 90% of individuals are EBV positive. A vaccine could modify the immune response to EBV and reduce risk of EBV reactivation in MS, but this is entirely speculative. We have the example of herpes zoster vaccine for the prevention of shingles, so that would be sort of the conceptual model. And there is also the theoretical issue of therapeutic vaccine in individuals who already have MS by modifying the immune response. Now, the role of EBV in driving MS activity remains unclear. So I think that this is an area in which we would like to get involved. Direct targeting of EBV as a way to better treat MS is a possibility, but remains speculative. Well, let me conclude saying this was the results of over 20 years of work. In fact, I started here, and I should thank Nancy Muller. In fact, our colleague Nancy Muller was the one who introduced me to the Department of Defense Serum Repository before the year 2000. This has been a unique collaboration with a military investigator that resulted in several landmark publications, including the demonstration that vitamin D level are an important predictor of MS risk, and several studies on EBV until we were finally able, I think, to bring this home with a more compelling evidence. And I want to thank here, the several people, but especially Sandy Munger, Sheetal Jarnevik, and Mariana Cortese were critical for the latest publication in Science. Our collaborator with the Uniform Services, University of the Health Science, and Brian Healy provided statistics support, and the team in Basel measured the serum neurofilament. So I hope I'm finishing time to leave time for questions. Looking forward to the debate. Thank you.
It's my great pleasure to introduce Dr. Cornelia van Duin, who received a doctorate in genetic epidemiology at the Erasmus University in Rotterdam in the Netherlands in 1992 and worked there until 2018 when she went to Oxford as a professor in the Nuffield Department of Public Health. Really great expert in dementia, Alzheimer's disease. Dr. van Duin will speak about the interplay between age, apolipoprotein E, and dementia. Dr. Cornelia van Duin.
Thank you, Albert. Can you see my screen?
Yes, but you have to put, yes, this is it. Thank you very much.
Thanks a lot. So it's a great pleasure to speak at this symposium. I know there was earlier one and I could not join, but this is fantastic presentations up till now. So I will talk about the interplay between age, apolipoprotein E, and dementia. And the background of the analysis is that if you look at dementia patients in the decade before the onset of disease, there are a large number of metabolic changes. So what we see in the patients, that brain glucose metabolism, at least in certain regions in the brain, goes down. And at the same time, although the utility in the brain is down, the blood glucose levels go up. Now, what we see at the same time, that patients in this period also lose weight and their blood pressure goes down. And that latter part, that the blood pressure goes down, is really known for decades now, that somehow the disease interferes with the metabolic condition of the patient before the onset of dementia. So at the molecular level, we have been studying also the changes in this period. And what we've seen is that there's certainly a very consistent decline in branched-chain amino acids, particularly valine and leucine. And we also see that the fatty acids, the PUVAs and the omega-3 are down consistently across studies. Now, it's a bit paradoxical, particularly the weight going down and the glucose going up. That is not what we would expect as epidemiologists. And this is one of the questions that has been bothering us over decades. And what we really know little of is what is the interplay between what is happening over age with the genes that are involved in dementia, as well as with the metabolic changes that we see. And what we basically lacked is any idea, is there a common mechanisms that explain these changes? Well, we think there is, and we think it has all to do with energy metabolism, which is a no-brainer as weight is involved. And we also start thinking that the player in that is not only age, but also apolipoprotein E. So the relevance of this, of course, is that if these are modifiable risk factors related to metabolism, they will be targets for intervention and improvement of the quality of life of the patient. So the study design is a follow-up study that is embedded in the UK Biobank. So we have about 125,000 persons characterized in the UK Biobank with the Nightingale platform for their metabolic profile. About 50,000 people are 60 years or were 60 years or older at baseline when profiled, and about 1,100 developed dementia at follow-up. 553 developed Alzheimer's disease and 298 vascular dementia. So the platform Nightingale is a platform that really targets cardiometabolic metabolites. we have measurements of 145 metabolites that are directly measured, and another 100 are inferred parameters. So it's the percentage of VLDL triglyceride over total VLDL. So these are just derived measurements that are calculated from these 145 direct measurements. So in the analysis that I will be presenting, we used a Cox proportional hazard model. We have about 10 years of follow-up of patients, and in these 10 years, patients have developed dementia. Of course, some of them were already on the road to dementia, but most of them are incident patients. At least half of them were developing during the period of follow-up. We adjusted for quite some variables that we suspected would be involved. That is ethnicity, age, sex, and BMI. Particularly BMI is a driver of these cardiometabolic measures. They involve cholesterols, inflammation, but also many diet-related metabolites like amino acids. Of course, then also fasting time is important. We further adjusted for more technical variables, assessment center and a batch and a spectrometer. And we adjusted for more vascular risk factors, smoking, alcohol intake, also education as a general risk factor for both vascular pathology and dementia. And medication, any medication that in an earlier study we had identified as being associated or interfering with the metabolite measurements. So we didn't do all at once. We had a model 1 which was quite restricted, but then we extended and looked at the number of metabolites that remained significant and didn't change sign, for instance. So if we consider the metabolites associated, we find a large series of metabolites associated with dementia at large. And some of them are quite consistent over categories, although often they are then significant in the case because we have twice as many Alzheimer's patients for Alzheimer's disease and not for vascular dementia. But there are a few exceptions. For instance, the small HDLs that are inversely related to vascular dementia and positively related are the large HDLs are significant mostly in vascular dementia and not in the larger group of Alzheimer's cases. So if we compare our findings to earlier studies, and of course this is a follow-up study with in 10 years after the basic measurement, but there's another study that we were involved in the epidemiological study that didn't include the UK Biobank yet. That's a study of Tinkin et al. It is about the same number of patients included, but of course our control or non-case set is much bigger in the UK Biobank. The findings are quite consistent. If you just look at the colors only, you see that the coloring is very similar in the studies. The point is that in the UK Biobank, we have more significant findings, partly explained by us using the FDR and Tinkin et al. Using a Bonferroni correction, which is more stringent. But overall, you can see that the findings are quite replicatable now over these two large follow-up studies, where I should say that the study of Tinkin et al. Is a meta-analysis of different population-based follow-up studies. What are the two new findings? Well, there's two sets. The HCLs that I already mentioned are associated with vascular dementia. And on the other hand, a series of metabolites, including citrate, beta-hydroxybutyrate, and glucose, but also other ketone bodies, that turned out to be very significant in the UK Biobank, but not in the earlier studies. So what is important to realize is that, of course, that these all these measurements on the cholesterols are extremely correlated. So you can imagine that all these subfractions of VLDLs HDLs, IDLs, and LDLs are very much correlated to each other. What you see from this picture is that it is mostly the HDL-VLDL axis that we find associated with the risk of dementia in 10 years. So not the LDL as such, that comes out with only one association. Most of it is the VLDL-HDL axis that is involved. What you also notice that if you look at the new cluster of the ketone bodies and the citrate, that is relatively not, it is significantly correlated to these lipid levels, but not as strong as these lipid levels, of course, are amongst themselves. These correlations amongst the lipids vary from 0.34 till 0.9, but the association of the ketone bodies and the citrate is much more modest. And also that of the branched chain amino acids is a little bit lower than the others, although stronger than for the citrate and ketone bodies. Now, as we are quite close to the onset, given the fact that we have only a follow-up of a little bit more than 10 years, we also decided to analyze these metabolites in those who did not develop dementia in the 10 years of follow-up, but associate then the metabolites to the same, to these outcomes on MRI, and evaluate whether the effect that we saw on either the MRI outcome or the risk of dementia, whether that was in the anticipated direction, and more or less confirming in a consistent way, suggesting that the metabolite also was involved in early pathology of dementia. So that gives us a very strong correlation. The p-value for the regression coefficient is 10 to the minus 14 for hippocampal volume, and total brain volume 10 to the minus 31. So this is a very strong correlation. In these pictures, you see the red line that denotes the significance findings for the dementia. And what you see that the beta-hydroxybutyrate and acetone comes up on the top side of the association. If you look at hippocampal volume, the findings are not significant, but there is a high correlation, and at least they showed an effect in the same direction. So these come also up, acetone, but also citrate comes strongly up if you look at total brain volume. And for both outcomes, we think that a smaller volume of the hippocampus or the brain is bad. So that should be inversely related to the association with dementia. What we also see for total brain volume is that the branched chain amino acids pop up as being associated, and these are a set of metabolites, the branched chain amino acids, particularly valine and leucine, that have been over and over again consistently associated to dementia. Also, again, negative correlation. Lower levels of valine are associated to a high risk of dementia. So we also looked at early pathology in terms of cognitive function, and that is not very well established as baseline in the UK Biobank. So we used reaction time as a measurement of more executive function. How fast can you react to a stimulus? Again, we expect, of course, the longer that takes you to respond, the worse your executive functions, and we find there very strong associations again in terms of p-value, but definitely these associations that we found for a couple of metabolites, including beta-hydroxybutyrate, the ketone bodies and the HDLs are very significant for both outcomes. And also on the other opposite, where also the branched chain amino acids and the VLDLs, HDL axis pop up. We don't find an association with fluid intelligence, which is a measurement of more general cognitive function or intelligence. And of course, one of the limitations is that we don't have an overview of a broad spectrum of cognitive function. So what one should realize is that apolipoprotein is the major driver of the nightingale metabolites platform. So what I try to visualize here in the ugly slide is that if you associate ApoE4 to the metabolites, there's only 4 metabolites in the platform that are not associated to ApoE4. You can do the same. So ApoE4, of course, increases the risk of Alzheimer's disease. ApoE2 supposed to decrease the risk of Alzheimer's disease. Again, very strongly associated with the metabolites that are part of the platform. Again, only 4 that are not associated. So it means that one thing to look at is ApoE, and what is also clear that if one looks at the aggregate of all the genes that are involved in Alzheimer's disease, so not only ApoE, but also the other genes that usually have a smaller effect than ApoE, you see that there is less evidence for association to the metabolites. And I haven't included a slide in this presentation, but if you look at what the effect of different genes is on these metabolites, they are often having opposing effects. So one is positive, one is negative. And what you see is a battle between the dementia genes in terms of effects on these lipids. What we were interested in is apolipoprotein because it's such a driver of the metabolites, and particularly in relation to the outcomes that we found were novel, beta-hydroxybutyrate, acetone, and citrate. And what we found, a little bit to our surprise, is that we see a quite strong effect, being modest in size, that the ApoE4 carriers on average over the lifetime have higher levels of beta-hydroxybutyrate. And the lowest levels are found in ApoE2, that is the protective effect, and the E3 are in between. So the E2 is protective for Alzheimer's disease. If one considers the effect of the E2 in general, it is a bit of a strange genotype because the 2,2s have a subgroup that has hugely increased levels of triglycerides, and this subgroup often shows early mortality. But if you survive as an E2 carrier, you often have a longer lifespan than the other genotype. So the interpretation of the 2,2 is easy for Alzheimer, but a little bit difficult if you look at cardiometabolic disease. What puzzled us here is, what does this mean? Because if you're an epidemiologist, you would say the straightforward interpretation is that beta-hydroxybutyrate increases your risk of dementia and Alzheimer's disease. And that surprised us a little bit because many of us are following diets or are knowledgeable of diets, and these are often ketone diets, where you try to increase your ketone levels in order to protect you from becoming obese, but also becoming diseased. Now, ApoE4 gave the same effect, of course, as age, because over age, from 40 to 70, your ketone, this beta-hydroxybutyrate ketone body goes up with age. So what we started thinking that this is a modification, that there's either pathology in the body occurring that explains this over age, the increase as a adaptive mechanism in the body to a disease that is emerging in order to have resilience and prevent damage towards the early pathology that is occurring. So that is well described and well acknowledged in metabolomics, that you have these resilience dynamics, adaptive mechanisms that you could pick up with these measurements. Now, of course, for those in clinic, what you start thinking of beta-hydroxybutyrate is starting thinking towards a mechanism that is well known, that in the case of the energy metabolism being disturbed, it is the task of the liver to generate ketone bodies, either beta hydroxybutyrate, but also acetoacetate is a ketone body that is exported into the circulation just to rescue your vital organs, the heart, the muscles, the kidney, and the brain in particular. So what the liver also will do in that case of an energy shortage is produce more glucose in order to help the organs that are in troubles. So well, that of course raised the question when we presented this, then people started putting the finger in the air and asking us, hey, doesn't this mean that there is just vascular pathology that is associated to a higher dementia risk that explains this phenomenon? So we did a lot of extra adjustment for pathology of the heart, kidney, sarcopenia, and lo and behold, everything stayed significant in this rather large study. Now, what is interesting that if you think about ApoE, of course, ApoE4 is also associated with longevity. And as such, ApoE is not very well, if you look at the mouse models based on ApoE4, it's not so much a model for dementia or Alzheimer's disease, but rather a model for aging. And if you look at aged mouse, then actually the ApoE4 carriers really show a higher vulnerability to bioenergetic deficits. And that is actually what we think we picked up with these beta hydroxybutyrate, but also with citrate. And citrate was a very important finding because citrate pointed us to what is known as the TCA cycle, because the high levels of citrate, which usually are high in the blood because generated by the osteoblast, but what you basically start thinking of that the citrate, the TCA cycle, again, is one of the mechanisms generating energy in the body. So we find that citrate is high. We find that the ketones are high. We find that the fatty acids are low. So we assume that these fatty acids are also burned in order to generate energy for the brain or other organs. We don't know for sure yet. But we also see that valine is low. So we also assume that that is burned in the TCA cycle just to generate more energy. And we find that glucose is high. Now, this combination, high citrate, high glucose, is really remarkable and points to troubles already as early as middle age. Now, does this also resolve the stunning question, why is glucose high in patients 10 years before the onset of the disease? Well, often the weight is low. And that's a bit strange because I think if you think about diabetes, if this were to represent diabetes itself, diabetes is associated with high branched-chain amino acid levels. So diabetes patients have high valine and leucine levels. So the fact that we find this strange combination rather may sense not so much that patients have diabetes as a disease, but that there is an energy metabolism in place that is generating as much as energy as it can in order to salvage the organs, including the brain. So we went one step further. So the valine levels, the amino acid, the branched-chain amino acids are really also part of this TCA cycle and part of the energy fuel that we have. And if we looked at the effect of ApoE on valine levels in the blood over age, we were a little bit even more surprised because again, you see here in green the ApoE tutus. You see in blue the ApoE4s, and what you see happening is actually that valine levels go up over age, so that is what you in line what happens in diabetes. But in the E4 carriers and a little bit later for the 3 carriers, you see that these with age these valine levels go down. Now, this is of course cross-sectional data, so I'm very much aware that this may be biased, and of course, particularly in the older age group, there has been bias of selection in the participants' inclusion, but at least it is an interesting finding because it mimics very much also what has been observed for BMI, and what we found for BMI, that the ApoE4 carriers go down over age in BMI already around between the age of 50 and 60. So what are we seeing? Check the time quickly. So what we think we are seeing in the blood is now the picture emerging that there is a problem with emerging in the energy metabolism, and that is really consistent with a role in which both acetylcoenzyme A plays a central role as one of the port entrees for the TCA cycle, as well as acetylcoenzyme A playing a key role also in the generation of these ketone bodies, beta-hydroxybutyrate and acetoacetate. So one of the questions is, of course, what happens in the brain? And we did that in a study in ADNI, in MAD. And what we find for Velen is not that there are low levels in the brain associated with Alzheimer's disease, but rather high levels. And that we see at the level of amyloid, as well as tangles, as well as other pathology, but we also see a positive association, very significant, given the small sample sizes, with cognitive decline. So what we basically see that in blood, the levels are low, and in the brain, Velen levels are high. For beta-hydroxybutyrate, we could not find any significant findings in ADNI. There is a tendency, however, that if you look at those with high amyloid levels, there is a tendency that every outcome, amyloid, tangles, other neuropathology is reduced in those with high beta-hydroxybutyrate levels. So if you combine the two, you get into the conclusion that there is in the blood low levels of branched chain amino acid, but there is a shift to high levels of Velen, at least in the brain, that we can measure. For the ketone bodies, we do not know. We really can't say this, but you could hypothesize that if anything we see that the ketone bodies are reduced, and that may, while they are increased in the circulation. So that, of course, raises the question, is that a problem of transport? And that brings me to the last part of the presentation, is that we don't know, and we don't know whether there's evidence even that ApoE is involved in transport, because we know a lot of functions of ApoE, and there's an overwhelming amount of function of ApoE, but nobody knows exactly yet how. So to resolve that question, we just ask ourselves, what is now the evidence if we knock out ApoE? What will happen then to the energy metabolism? And this is a study that we used the data for an IMI project that I was involved in, Adapted. And what we basically generated in this study from two donors, a person who was a 3/4 carrier and an other person who was a 4/4 carrier. The 3/4 was a healthy person, the 4/4 was a patient. And what we basically generated for these persons is iPSC, and we started editing those lines. I will not show that data. I will show them for the original genotypes of the patients, and made, based on the iPSC, differentiated macrophages, microglia, astrocyte, and neurons. So my question was, like, the hardest evidence that we can get that ApoE is involved in energy metabolism is just knocking out the gene of the original and see what's happening then. Well, if you knock out a gene, whatever you do with these genes, there's always effects. So we wanted to see a consistent effects in these two work streams lines. So in the two persons should show a consistent effect over the effects of knocking out ApoE. And actually what you see quite consistently, and we do not know whether it's biased, but the very mitochondrial gene CHCH-D2, which is a rare Parkinson involved gene, shows the bigger effects of it, even huge, bigger than ApoE itself in microglia. And you definitely see in microglia an effect on a solute carrier that is involved in the transport of coenzyme A and TCA related metabolites. You see even a bigger effect in the astrocyte. So the astrocyte is the cell in the brain that is ketogenic, together with pericytes, that are generating ketones in case of energy deficiencies in the brain for the neurons to survive. And in the astrocyte, you really see different solute carriers popping up, as well as, again, the CHCH-D2. So summing up, I think I'm 2 minutes late, but sorry for that. I think we have a study that suggests that energy imbalance may explain the metabolic changes we see in patients in the decade before the onset of the dementia. That is, the TCA cycle is upside down, and both the coenzyme A is really involved. We find evidence that ApoE is driving that, and that ApoE may change over the life course. Of course, we have to replicate this really in follow-up settings, but we also see that ApoE is involved in energy metabolism in iPSC. So our hypothesis is that the liver compensates the energy deficits in the brain as early as midlife, and that may have implications to improve, first of all, the general health status of a patient in that period before the onset, but also the quality of life, and maybe also the chances of success of trials. If you are trying to do a trial in a patient that is in an energy deficit, I think the chances may be reduced to have a positive outcome. With that, the thanks to the people who did the work, John Liu, of course, and Najaf Amin, who really made all the analysis happen, and then all the advice and interest of Rima Kadura-Duk, who was the co-leader of the study, and a lot of statistical input from both teams, both in Oxford and at Duke and Germany. With that, thank you for your attention.
Thank you very much. I know there are a few questions, but I would hope we keep them to the end, because in the sake of time, I would like to go now to our third speaker, Thomas Littlejohns. Thomas is a senior epidemiologist in the Translational Epidemiology Unit at the University of Oxford. And Thomas, it's wonderful to have you, and you will speak about hearing impairment as a potential modifiable risk factor for dementia. Dr. Thomas Littlejohns, please go ahead.
Yep, thank you for the introduction. I'm just going to share my screen. If you just let me know if you can see it. Is that?
Yes, this
yep.
is it. Yep.
All right,
thank you very much.
Brilliant, perfect. Yeah, so the focus on my presentation is, so just a brief bit of background. My interest is focusing on modifiable risk factors for dementia, and in the last few years, I've been focusing on sensory impairment. So this presentation is going to focus specifically on the role of hearing impairment. And if you're familiar with risk factors for dementia, then you might be aware of this paper, which came out in 2017. So it's quite influential in terms of kind of highlighting, I think, the potential role of hearing impairment. So this paper by Livingston et al., which was published in The Lancet, it was essentially a commission that was focusing on just providing an overview of risk factors for dementia. And just in this figure, so they highlighted 9 modifiable risk factors and Many of these are pretty well established. They're common risk factors for other diseases, but things like hypertension, smoking, diabetes is touched on in Cornelia's presentation. So these are pretty well established, but what kind of caught the attention of a lot of people is that they included hearing loss as a risk factor. So it wasn't just the fact that it was included, but actually on the far right hand side column, they provide the population attributable fraction of these risk factors. And actually out of the 9 that they highlighted, hearing loss had the highest population attributable risk fractions, so about 9%. So essentially they're saying that assuming causality, that 9% of dementia cases will be prevented if we addressed hearing loss. So this is based on the relative risk. So there's a strong relative risk of 1.9, but also hearing loss is pretty high prevalence as well in the age group they identify. So around about 30% of individuals who are middle-aged have some form of hearing loss. But I think when you take a bit of a closer look at the figures underlying this, so as I mentioned, some of these are very well established. There's a strong research literature going back many years in terms of the role of these factors as risk factors for dementia. Whereas in this meta-analysis they present for the relative risk, this was based on actually 3 longitudinal studies. So in these studies, they had fairly long follow-up. So about the range was 9 to 17 years of follow-up. So hearing loss and association with risk of dementia followed up 9 to 17 years later. But the sample sizes were fairly small, so 600 to 2,000 participants. And the total number of cases was, the highest number of cases in these studies was 229. So essentially, there's a lot of interest in this role as a risk factor, but if you think about the terms of the evidence base, there clearly does need to be more research in this area, in particular, just looking observationally at the associations between hearing and risk of dementia. So this commission was 2017. They published an updated version in 2020, expanded the list to 12 modifiable risk factors. But again, hearing loss was the risk factor with the highest population attributable fraction. And I think the meta-analysis, I don't think they updated it. So I think it was still based essentially on these 3 studies. So just briefly to think about the kind of mechanisms that might underlie this association. So these are, given the novelness of this as a risk factor, this is still fairly kind of hypothetical, but essentially it could be causal, and you could split it up maybe by direct or indirect. So direct, that is sensory deprivation within the brain itself. So obviously you have sensory regions in the brain, perhaps having atrophy in these areas produces some kind of knock-on effect. Indirect could be feasibly through other factors. So this is something that's been underexplored in previous studies. So essentially, people who have developed hearing problems in middle life, maybe they become more socially isolated, maybe they develop, depression or other risk factors for dementia. So there could be this kind of indirect pathway where hearing loss is influenced in other risk factors for dementia. And of course you have not so much pathways, but kind of explanations for what might be going on for this association. So confounding is the most obvious one when we think of observational studies, but reverse causation as well, in particular for dementia. So Cornelia really touched upon this in the last presentation, but with dementia, it's really difficult to disentangle temporality, because even if you have the exposure, even in a longitudinal study where you have the exposure measured prior to the development of dementia, that's usually clinical diagnosis of dementia. So dementia has this long prodromal preclinical phase where it can take place over many years. And in a way, this touches upon confounding again. So that prodromal period can influence health and other factors, which could then influence, for example, hearing and produce essentially a spurious association. So we kind of looked at this question. We tried to address some of these issues. So really what we wanted to do was actually just look at the association and just do it in a bigger sample size and get an idea, what the association looks like. So again, we used UK Biobank. our sample size consisted of just over 80,000 participants. They were aged between 60 and 69. So the exposure that we had measured was a form of hearing, so it's speech and noise. So participants went into these baseline assessment centers. They took a hearing test. So this involved them doing a test of their ability to detect speech and noise. So they did a digit triplet test. And then we categorized these participants based on predefined categories based on previous studies. So around 20% of the participants were categorized as either insufficient or poor hearing. And over the course of around 10 years of follow-up, so a median of 10 years of follow-up, there were almost 1,300 cases of dementia developed in these 80,000 participants. So you kind of see in the plot in the right-hand side, just presenting the raw numbers, that between those who are poor, insufficient, and normal, they're kind of starting to spread out after around 2 years of follow-up, with those with worse hearing going on to the incidence of dementia, the rates is higher in those who have worse hearing. So we used a Cox proportional hazards model adjusted for the variables that I've listed on the slide, and we kind of confirm what we see in that plot. So we see kind of a dose response association. So those with insufficient hearing compared to normal, hazard ratio is 1.6, and those with poor hearing, the hazard ratio is 1.9. And the 1.9 is quite interesting because actually, if we think back to the first slide from the commission, the effect estimate they provided from that meta-analysis of 3 studies was also 1.9 exactly. So this is quite similar in terms of the effect size. And then on the right-hand side, so we try to disentangle this element of reverse causation. So again, it's very difficult to do unless you have even longer follow-up, but we segmented the follow-up periods into 3-year bands. We kind of find 2 things. So firstly, so dementia cases that developed around 9 years later, the effect estimates were pretty consistent with the main findings, although you can kind of see they do attenuate throughout the follow-up period. So, if you're hypothesizing that reverse causation might be going on, then you'd expect a stronger association within the first few years, and then this sort of decline over a follow-up period. But it does still remain after around 9 years, similar effect estimate. And then we try to look into some form of mediation. So we looked at whether depressive symptoms or social isolation, or both these factors combined, if there was any evidence that excess risk could be mediated by these conditions. So if we just focus on the number right at the bottom of this figure on the left, so the excess risk mediated by having both depressive symptoms and isolation, this sort of suggests that maybe a little bit might be mediated by these factors, but only a very small amount. So I think this is something that could be explored a little bit further. But just thinking about the indirect pathways, maybe these factors aren't a strong driver of what might be going on in terms of the association. And then we also had information on hearing aid use at baseline. So when we actually stratified by whether participants wore a hearing aid or not, then we see that actually in those who don't wear a hearing aid, the association remains pretty consistent. Whereas in those who do wear a hearing aid, it's attenuated quite a bit. Just to note, we do have quite small numbers, and I think this is kind of, again, highlighting, the advantage there would be of even bigger data sets, or data sets enriched for participants who have, more kind of forms of hearing impairment, because even though we have such a large sample size, we are still getting into quite small numbers when we look at some of these questions. And then just a final result, we wanted to look at self-reported hearing impairments as well, because actually a lot of the literature, so that meta-analysis I mentioned at the beginning, so that was based on 3 studies that had an objective measure of hearing impairment. There is quite a bit of literature that has looked at self-reported hearing impairment, and they tend to find this kind of increased risk as well. So in UK Biobank, participants did report whether they had some form of hearing impairment. And I think this is useful as just a general indication of what the risk would be in these participants, given that with speech and noise, we are capturing a very specific component of hearing impairment. So So we find an increased risk in these participants, not particularly strong hazard ratio, but almost a third of participants report some form of hearing impairment. I think what's quite important to consider is that, looking at this crosstab of those who say they have a hearing impairment and then those who actually are classified as having poor hearing based on the speech and noise test. So those who have, say, a normal speech and noise hearing, 30% of those self-report hearing impairment. I don't think that's too surprising because, of course, you could have other forms of hearing impairment and you might not really even consider the role of hearing, speech and noise. But amongst those who perform poorly on the speech and noise test, which is around 3-4% of participants, whilst 2/3 do say they have poor hearing, a third we're not capturing with self-report, so we might just be capturing a little bit of information that people don't necessarily consider as poor hearing. So just to summarize, so we kind of replicate what's been found in previous observational studies. Slight distinction being is that we're focusing on a specific component of hearing, that is speech and noise measured hearing. There are obvious limitations where you have in this kind of study. I just wanted to focus on two more broader limitations, particularly as people who are watching might be interested in using UK Biobank for dementia research. So in terms of the way dementia is ascertained at the moment, it's based on hospital diagnosis or cause of death on death records. It's primarily through hospital diagnosis. So there's kind of two elements to that. First is that will obviously under ascertain cases. That said, there has been research, comparing that method of diagnosis with other medical records, and they showed that these are likely to be true positive. On the whole, these are likely to, be valid for capturing true cases, but they will under ascertain cases. And another element of that is capturing the later stage. So if we're thinking about the role of reverse causation, then we're capturing dementia when they're at the severe end and getting captured in, in hospital or on the death certificate, whereas these individuals might have been diagnosed or might have developed dementia clinically in primary care a couple of years prior to that. So that's just another thing to consider. So that kind of affects the follow-up. So maybe we need even longer follow-up than 9 to 11 years to really kind of get into this reverse causation issue. So Cornelia mentioned the findings that she had for the imaging, and I think that's where a strength of UK Biobank really comes into for dementia research as well. It's not just the sample size. We have UK Biobank has data on currently 50,000 participants have been imaged, with around 4,000 who've had repeat imaging as well. So you can kind of look at change over time. And the aim is to increase those numbers to about 100,000 to have some form of imaging, and 70,000 with repeat. So we can start looking at the associations several steps before that clinical onset. And I think in the context of hearing, particularly as you could feasibly have changes in the brain due to dementia, which also affects the ability to hear properly as well. So that reverse causation hypothesis again, we can start disentangling that a little bit more using the imaging data. So I think that would be an interesting next step to follow up these findings with dementia.
Thomas, thank you very, very much.
And yeah, my acknowledgements. Thank you.
Go to our final presentation. Dr. Tianxian Ye has medical degrees from University of Taiwan, National University of Taiwan, a PhD in clinical epidemiology at the Harvard Chan School, and is currently a postdoctoral fellow in the Harvard-Oxford program in epidemiology. And she is affiliated with the Department of Epidemiology of Harvard Chan. It's a great pleasure, Tianxian, to give you the floor. You will speak about long-term dietary intake and subjective cognitive decline. Go ahead.
Yes, thank you so much, Professor Hofman, for the introduction. Can everyone see my screen?
I can, yep.
Okay, great. Yes, so it's a great honor to be here. Today, I will be sharing the results from our recently published paper on dietary flavonoid intake and subjective cognitive decline, or SCD. Dementia is an important global health challenge, and diet, as one of its modifiable risk factors, has received growing attention. The course of dementia is a continuum of decline from normal cognitive function through a preclinical phase, and then objective cognitive impairment, and finally dementia. The outcome of the current study, the SCD, is a stage when there is self-perceived cognitive decline, but the cognitive impairment still cannot be detected during formal testing. The SCD was found to be strongly associated with both concurrent objective cognitive function and subsequent cognitive decline. And it may be especially important in people who received higher education. The main exposure of this study, the flavonoids, they are naturally occurring phytochemicals found in many fruits and vegetables that we consume. According to their structures, they can be classified into different subclasses, and they've long been considered as powerful antioxidants. Because oxidative stress may play a role in the pathogenesis of dementia, so flavonoids have been proposed as potentially effective agents for cognitive decline prevention. But studies on the associations between flavonoid intake and cognitive decline have remained inconclusive. Therefore, we use data from the Nurses' Health Study and the Health Professionals Follow-up Study to investigate this topic. We have a total of more than 77,000 participants included in our final analysis. And during the more than 20 years of follow-up, we have multiple dietary assessments and 2 SCD measurements. We stopped updating the dietary information 6 years prior to the outcome assessment to minimize reverse causation. Our dietary data was from the food frequency questionnaires, and we focused on these 7 flavonoid subclasses, which are commonly consumed in the Western diet. The SCD was based on these 7 questions, and 1 point was given for each yes. So 0 is the perfect score and 7 is the worst. Because 3 or more positive SCD questions have been used to indicate poor cognitive function, so we calculated odds ratios for 3-unit increments in SCD. We adjusted many potential confounding factors in our models, including non-dietary factors and dietary factors such as vitamin C, D, E, and fatty acid intakes. Now on to the results. These 2 figures are showing the multivariate adjusted odds ratios for each flavonoid subclass by quintile in the both cohorts. You can see that almost all the flavonoid subclass have protective associations with SCD. When we combine the results from the 2 cohorts, the strongest association was observed for flavones, followed by flavonones, and then anthocyanins. We also looked at the dose-response relationship between flavonoids and SCD. The x-axis is the absolute intake amount, and you can see that the curve was the steepest for flavones, followed by anthocyanins. These findings may be helpful for future interventional studies. We then explored the food level and found that many flavonoid-rich foods had protective associations with SCD. We used stepwise regression and found that strawberries, blueberries, peaches, citrus fruits and juices, and apples were independent predictors of subsequent SCD. For example, the pooled odds ratio for every 3 servings per week increase in strawberries was 0.58. We noticed that citrus fruits and juices, berries, and apples were top food contributors for flavones, flavonoids and anthocyanins in our cohorts. Therefore, we can say that our findings on the food level further support our findings on the phytochemical level. We also investigated the temporal relationships of flavonoids with SCD. Take flavonoids, for example, you can see that the inverse associations were consistent throughout the 20 years of follow-up, and also when we included both recent and remote intakes in the same model. These findings may suggest that it's never too late to start incorporating flavonoids into your healthy diet, and that the associations we observed may be less likely to reverse causation. And similar temporal relationships were observed in both cohorts and also on the food level. Some possible mechanisms of flavonoids on cognitive function have been proposed, including reduction of neuroinflammation, oxidative stress, amyloid level, and also increased neurogenesis and neurotrophin production, etc. So the conclusion of this study is that we found flavones, flavonoids, and anthocyanins had the strongest protective association with SCD, and many flavonoid-rich foods, such as berries, citrus fruits, and juices, may be beneficial in maintaining cognitive function. Another related paper that we recently published is about protein intake and SCD. And in this paper, we found that higher protein intake was associated with lower odds of SCD when compared with total carbohydrate, and that plant-based protein was the superior source compared with animal protein. And also the food sources may also be important. Among the 9 food groups, 9 protein food groups that we examined, we found the strongest protective association for beans and legumes, followed by lean chicken and fish. And we also noticed that processed meat products, such as the hot dogs, were associated with higher odds of SCD. In the end, I would like to express my sincere thanks to all my mentors, Professors Deborah Blacker, Walter Willett, Alberto Escurial, Bernard Rossner, Mark Weiskopf, and David Hunter, and all my wonderful classmates and colleagues and friends at Harvard and also Oxford Translational Epi Unit. Thank you all very much for listening.